All right, everyone. These are the lesson 9 group problems. You can see here are number 1. You can see all the things that were required. We have here the origin. We have the origin here. Sorry, and then this is actually cut in half. So you can see here there's the leading strand, and it'll continue on the other side as a lagging strand. And then on the bottom here has the lagging strand, and continuing to the other side would be the leading strand as well. Here we have DNA polymerase 1 that will change RNA primase to DNA. We have RNA primase at the head of each DNA that's getting replicated. There's the DNA polymerase 3 alpha subunit that replicates the DNA. Here we have Okazaki fragments right there. Here we have another DNA polymerase 3 subunit, alpha subunit. Here's DNA helicase, topoisomerase 2, and the black strands are the parental DNA.
All right, so number 2 is just find the terms. So we got primase, this protein complex synthesizes primers in eukaryotes.
DNA is the protein that promotes the separation of the AT-rich sequences within ORIC.
Epsilon is a subunit of DNA polymerase 3 contains proofreading activity.
And then the epsilon subunit of DNA polymerase 3 is the protein that synthesizes the leading strand in eukaryotes.
Termination utilization substances. This protein displaces DNA polymerase 3 from the chromosome to terminate replication.
And DNA polymerase 1, this protein has a 5-3 polymerase and 5-3 exonuclease activity.
DNA diarase, this protein includes negative superpooling into the DNA and other replication form. It's also called
Topoisomerase.
Topoisomerase type 2.
The origin recognition complex is the protein that binds directly to the ARS elements.
Single-strand DNA binding proteins. This protein provides DNA strands for healing during replication.
And DNA ligase is the enzyme that seals Okazaki fragments into a continuous strand of DNA.
All right, so number 3. So the list of 5 enzymes, that's DNA helicase, DNA diarase, DNA primase, DNA ligase, and DNA-A. And then for list of 5 enzymes that do during the GTP, DATP during DNA replication. So this one is a little bit confusing, but there's a DNA polymerase 1 and 3, and there's also the subunits that we included in there because we could find anything else. So it's the epsilon, alpha, and delta. And then list 5 is just the same, that do the 3 to 5X nuclease. And then indicate that one enzyme involved in replication that includes DNA as a template to synthesize RNA, that's DNA polymerase. Include one enzyme that uses RNA as a template to synthesize DNA polymerase 1. That's what we found.
All right. For number 4, we had to take that DNA strand and take off the primer part of it. And so after you do that, for part A, you have these DNTPs that are here, and all of them are good. So you'll be able to just replicate the entire strand. And so it should be ACT, GAT, GGT, CCT, GCTAT. And then for part B, you can see here that there's a DCMP, and that one can't be replicated, can't be used in replication, in translation, I mean. So it's going to stop after one nucleotide at A. This one for C uses UTP, and that one will stop translation as well. So it stops after 2 nucleotides, and that's AC. And then this one uses GTP, and it's not deoxy GTP, so it can't be used for to translate DNA. It's for RNA, so it'll stop after 3 and it'll be ACT.
Okay, so this one's just finding which one's, a part of this. So for prokaryotes, there's the DNA virus, the TUS, the DNA A, DNA polymerase epsilon, DNA primase, DNA polymerase 1, DNA polymerase alpha, DNA polymerase 3, alpha subunit, and PCN. Okay. All right, and then for the mouse, so mouses are eukaryotes, so you want to remember that when you're reading which ones there are. So it's not just all of them. You want to exclude the DNA A because that's part of just prokaryotes. It's for the replication process for that. So it's all but that one. And then which of the above proteins include in the chromosome compaction? So that would be the CDC6, MCM helicase, DNA A, and polymerase.
Primase.
Primase. Which one of the proteins is involved in the initiation stage of DNA replication? So that would be the same ones. They help with that initiation, finding, the origin, replication swap. Okay. Which of the proteins is involved in the termination stage of DNA replication? So that's DNA virus, the TUS, and the PCN.
All right, hope that was helpful and that you guys enjoyed it. And we'll see you later.